the other one. Okay. So good afternoon, dear students. I invited a colleague from Germany, and so you will have a seminar next 7th April. And this colleague is a very important figure in Germany of medicinal chemistry. And so I think we make a seminar of about 1 hour about the research in medicinal chemistry in English. So you have the possibility to hear something, a scientific report, directly from a scientist. And so during our lesson. The name is Stefan Laufer from Tübingen. Tübingen is an important university in Germany. So I have already sent to Caterina the curriculum of this scientist, and so perhaps you can see.
I sent that.
Okay. Thank you, Caterina. Grazie. And so I think it's a good thing for you to start to hear something, somebody about the research in English without any problem for me. So, okay, it's really a pleasure for me. Okay, so for today we have to finish the introduction part, only 2 concepts, and then we start with the specific topics, and in particular about chemotherapy. And so today we have to discuss something about antibiotics and so on. At the end of this introduction part, there is an important concept that I'd like to introduce you. In particular, is the concept of a pharmacophore. You see, and just a moment. Okay. A pharmacophore, we can read together the definition, is defined as the tridimensional arrangement of features that is crucial for a ligand molecule in order to interact with a target receptor in a specific binding site. Pharmacophore is the minimal chemical structure needs to have a specific interaction with a specific receptor. You can see here in this example. On your left, you can see the chemical structure of heroin. It's a complex structure. You can see 5 rings fused together, basic nitrogen atom, 2 esters, very big molecular weight. But the problem was, what is the part of this molecule responsible of the interaction with the morphine, heroin receptor, morphine better, receptor. Scientists worked a lot in the past, and they found it is sufficient to have a basic nitrogen atom, a linear chain, and an aromatic ring. Following this approach, they discovered the structure and the activity of methadone. Methadone is a very simple structure. It's possible to synthesize this compound in a lab without any problem. And methadone has the same biological activity of heroin. And so you see, heroin is a natural, morphine, better, is a natural product, can be transformed in heroin, but we need to have opium from Papaver somniferum, and sometimes we have no quantity of this compound from agriculture. By applying this approach, it's possible to synthesize methadone in a large scale, big quantities, and so we can use this compound without any problem. Here on the top you can see the chemical structure of the pharmacophore for this type of receptor. We'll see this concept, we'll see this approach directly on the chapter of this compound. So when you have a molecule, we can distinguish three different parts. The first one is the pharmacophore. We have already defined, is the chemical structure, the minimal chemical structure necessary to have a particular biological activity. But there are also other two important parts, a toxicophore and a metabophore. Toxicophore is the part of the molecule responsible of specific toxic effects. Sometimes there are particular substituents on the molecule that can be transformed by our metabolism in toxic group. And it is important to know toxicophore. Why? Because we can try to transform it in a non-toxic group, maintaining the same activity. By applying the isosterism, we have already seen how we can replace some atoms with other atoms, trying to maintain the same biological activity. And finally, there is the third part of a molecule called the metabophore, is the three-dimensional set responsible of the metabolic properties. So, in this case, if we want to prolong the activity of a specific molecule, we can investigate, we can try to modify the metabophore in order to increase the resistance of the molecule to the activity of particular enzymes and so on. So three parts in the molecule: pharmacophore, toxicophore, and metabophore. All the rest is defined as baggage, luggage, what you want. You can see in the picture, in green, the pharmacophore, necessary for an optimal interaction with the receptor, and the other two parts, the metabophore and the toxicophore. If we want to understand better understand what is a toxicophore, we can see this example related to an old anti-inflammatory drug called thalidomide. Thalidomide was used in the past after the Second World War, and it was a potent painkiller. It was used by the people, the population, but it had in that time severe problems. You see the chemical structure of this compound, and you can see this chiral carbon atom. Only the air enantiomer is active as a sedative. And so the compound was commercialized in air form. But in vivo, this carbon atom can be modified, you see, and we can get the S enantiomer by our metabolism. And the S enantiomer is teratogenic. The S enantiomer was responsible of the children without arms. And so if pregnant women used this type of compound, in that time she had problem to have a child without arms, teratogenic. And so you can see this part of the molecule on your right is considered the toxicophore because the modification of the chiral center is responsible for the production of the teratogenic enantiomer. If you see on the web some picture about the use of this drug, you can find a lot of picture of children without arms with a lot of problems. So we have to avoid the presence of this chiral carbon atom if we want to avoid this type of side effects. Now we have the last point of the introduction part, enzymatic inhibition. During the course of biochemistry, you have seen a lot of concepts, a lot of description about the activity of the different enzymes in our body. Enzymes are important, yes. They can decrease the activation energy, and so they are necessary if we want to perform some reaction at a body temperature, 36, 37 degrees. But sometimes we can inhibit their activity to have a particular therapeutic effect. Here in this table, you can see a series of different enzymes, and you can see a series of inhibitors of these enzymes. These inhibitors are used as drugs because, as I told you before, it is important sometimes to block the activity of particular enzymes if we want to have a particular therapeutic effect. One of the most famous examples is acetylcholinesterase. You know that this enzyme is present in the neuromuscular junction, and these enzymes are important to block the contraction of the muscles. But sometimes we need to inhibit this type of the activity of these enzymes if we want to increase the quantity of acetylcholine in the synaptic cleft. It's the case of myasthenia gravis. Myasthenia gravis, it's a severe disease in which there is the formation of particular antibodies against the acetylcholine receptors. In this case, this type of patients have a problem to contract their muscles. And so it is necessary to increase the quantity of acetylcholine in the neuromuscular junction. It's possible if we block the activity of acetylcholinesterase. And so you can see neostigmine, ecothiopate, and so on are acetylcholine inhibitors used as a drug to treat this type of disease. Here, there is another interesting example, beta-lactamase. The next chapter of our course is related to penicillin, antibiotics. And you know that there are several strains of bacteria resistant to penicillin because they have a particular enzyme called the beta-lactamase able to open the beta-lactam ring. We'll see in the next chapter the reactive beta-lactam ring of penicillin. If we use clavulanic acid is an inhibitor of beta-lactamase, and so clavulanic acid is able to block these enzymes able to open the ring of the penicillin. And so we block these enzymes and we can use a normal penicillin because the beta-lactamase are blocked and the normal penicillin can reach the target and to kill the bacteria and so on. So these are two examples of enzymes inhibitor used as drug for the therapy of particular diseases. Okay. You can see that we can have a reversible inhibition. It's an equilibrium, so we can have competition between the substrate and our inhibitor. Or we can have also a non-competitive inhibition. In this case, the inhibitor binds to sites outside the catalytic site. We can have also irreversible inhibitors, and in this case, this type of inhibitors are able to give a covalent bond with the active site of the enzyme or with a cofactor. You know that normally an enzyme has a particular cofactor necessary to have the activity of this particular enzyme. The reversible inhibitors are divided in two groups. The first one are inhibitors at the active site. So we have the active site, substrate normally interact with the active site. We can have a molecule able to occupy the active site of the enzymes, and in this way is able to block the activity of this particular enzyme, giving a covalent bond with the active site. There is a second group of inhibitors, irreversible inhibitors, called suicide inhibitors. Why this term? Because this type of compounds normally are not active. So we can use this compound, but they are not active. they can give their particular activity only after their interaction with the active site. So the activity of this compound is activated, you switch it on what you want by a reaction with the particular group present in the active site. You can see here an example so you can better understand this concept. This is the chemical structure of a penicillin. You can see this ring, very small, 4-member ring. We'll see this ring, we'll see the chemical properties of this ring. It's a very reactive ring. It's a potent electrophile, can be attacked by nucleophiles present in the active site. And so in this case, you can see that this group, this OH, this alcoholic group of a particular serine residue located in the active site of these enzymes called transpeptidase. And this OH, this nucleophile is able to attack the electrophilic carbon atom of beta-lactam ring. And so we can observe the formation of a covalent bond between the OH of serine and the activated carbonyl atom of the beta-lactamic ring. So in this case, you can see that penicillin normally are not reactive, but only after the interaction with OH, they can block irreversibly this type of enzyme. Here you can see another type of reaction used in the case of suicide inhibitors. We have already seen this type of reaction called Michael addition. Michael addition is given by unsaturation very close to the carbonyl group, alpha beta unsaturated ketones normally. And this type of carbon atom can be attacked by nucleophiles. Here you can see a cofactor present in the monoamino oxidases, particular enzymes able to oxidize monoamino neurotransmitters like serotonin, noradrenaline, and so on. And you see the flavinic group, the cofactor, as a nucleophilic nitrogen atom. Here you see this type of atom as a minus charge. And this minus charge is able to attack the Michael acceptor. The Michael acceptor is a drug called the pargilin, and you can see the formation of a covalent bond, and in this way the enzyme is blocked irreversibly. This is another example of suicide inhibitors, in this case pargilin. Pargilin can block the activity of this particular enzyme. There is another important point. When we want to design new inhibitors of a particular enzyme, how we can know the starting point, how we can know the chemical structure, how we can design a particular inhibitors of a particular enzyme. If it's possible, we can start from the transition state of the enzyme. You see here in the picture reactants, transition state, and products. Normally, the transition state has the same chemical feature of the starting point and the same chemical features of the products mixed together. If I am able, you know that the transition state is not stable. But if we stabilize this type of molecule, we can have an optimal inhibitor of a particular enzyme. Here you can see a practical example, for me very clear. In the viruses, there are particular enzymes called proteases. Proteases are important enzymes able to hydrolyze the peptidic bond in particular points to give small peptide that should be assembled by the virus to form new virions. In the virus of HIV, it was observed a particular hydrolysis between the phenylalanine residue here and the proline here. So you can see the peptidic bond here. And it was observed a particular hydrolysis of this bond given by an aspartic proteases. Aspartic proteases is a very uncommon enzyme in mammalian. And these enzymes has two arms or two acidic residues of two aspartic acid residues. There is also a water molecule in the active site, and so you can see the formation of this diol, geminal diol. Geminal diol is not so stable. It's an hydrated form of a ketone, not stable. And you see that after the formation of this diol, you can see the hydrolysis of the bond, of this peptidic bond. So the scientists tried to design a stable, quite stable alcohol just to mimic this type of molecule in order to have a selective inhibitor of this type of enzyme. Here you can see the scientist designed this particular hydroxyethylene bond in which you can see a nitrogen atom here, the alcoholic group here, another nitrogen atom here, and this particular structure represents the pharmacophore of several inhibitors of this class of enzymes, proteases, saquinavir, ritonavir, indinavir, and so on. So it was possible to design this type of inhibitor starting from the chemical structure of the transition state of this reaction. Once again, you can understand the importance of the transition state of a particular reaction if you want to design inhibitors of this type of enzyme. The last point, the last strategy to inhibit the activity of particular enzymes, it's given by the design of antimetabolites. Antimetabolites. This term means that we can start from the chemical structure of a substrate and we can introduce some small modification following the rules of isosterism, just to have a molecule very similar to the substrate with the same electronic volume, for example, but with small chemical differences inside their structure. You see here an example. Folic acid. Folic acid, it's very important for the cell life because it is a donor of carbon unit. Folic acid is important in the synthesis of DNA bases. So if we block the formation of folic acid, we block the formation of DNA for a cell. In the past, some years ago, the scientists designed an antimetabolite very similar to folic acid called methotrexate. You can see the chemical structure is quite the same, but with a small difference. In the folic acid, you can see here an OH group, enol, and the OH group, following the rules of isosterism, can be replaced by an amino group, same volume, but you know that amino group is quite different from an alcoholic group. Amino group has a ion pair, so amino group is able to increase the basicity of this heterocycle, in particular of this nitrogen atom. This nitrogen atom can be protonated at physiological pH, and so we have a strong, the final result is a strong interaction of methotrexate with the active site of a particular enzyme called dihydrofolate reductase. We'll see this chemical modification, we'll see the activity of methotrexate as anti-cancer, but now it is important that you understand that it's possible to introduce a small modification inside the chemical structure of a substrate in order to have an antimetabolite. Here you can find the definition of the antimetabolites. Are drugs that interfere with one or more enzymes or their reactions that are necessary for DNA synthesis. But I want to show you another example, and we'll see also this example in a future chapter about anti-cancer drugs. Here you can see a biochemical pathway We start from uracil, and the final product is thymine. There is only one methyl group. You see, the difference is only in one methyl group. So there is a specific pathway able to introduce a methyl group on the backbone of uracil. What happens if we change the chemical structure of uracil by introducing a fluorine instead of an hydrogen? You know that fluorine is very small. Fluorine has the same electronic volume of an hydrogen atom, but you also know that fluorine has a completely different chemical property. In this particular pathway, you can see the exit of a proton, of an hydrogen like a proton. So it is necessary, the exit of this proton, to have this double bond and then to have the addition of a methyl group. If there is a fluorine atom instead of an hydrogen atom, we should have an exit of a positive fluorine atom, F+. And you know that normally an halogen has a minus form. It's not positive. It has not a positive charge. And so you can understand that if we use fluorouracil instead of uracil, we have the formation of a covalent bond of the fluorouracil with the cofactor, and the reaction is stopped because it's impossible the exit of a positive fluorine at this level. So these two examples are useful for you just to understand the strategy. Small modification of the molecule of the substrate to block important pathway inside the cells. In this case, we can trigger the apoptotic pathway, and so we can use fluorouracil in the cancer cell if you want to kill the cancer cell. A third example, it's a well-known example. This example represents the first example of an antibacterial agent used in the past. Sulfonamides. You can see here the chemical structure of a sulfonamide. The chemical structure of this compound is very similar to the chemical structure of the substrate, para-amino-benzoic acid. You see the dimensions are more or less the same. Also, the chemical properties are more or less the same. Anilino group here, acidic group. Sulfonamides has an acidic proton, the same of the carboxylic group. And so these two molecules are very similar. And so in this particular pathway, you see, it is important, the incorporation of a para-amino-benzoic acid, to have the folic acid. This important molecule able to provide methyl group, to introduce a methyl group in the uracil to give thymine. But if we use a we take sulfonamides, sulfonamides can be taken by the cell, and you see the pathway is blocked at this level. So once again, we can perform small modification in the molecules of the substrate to have antimetabolites. So, have you any questions about this part? Also at home, have you any questions? No? Okay.
It's clear, Prof.
Okay, thank you, thank you. So, we have finished the introduction part now, and we can start with the next chapter on antibiotics. Okay, thanks. Okay, we can close this chapter. Just a moment. Slides, data. Okay. Try to see on the Teams if I see the slides. Just a moment. Oops. Shut. Okay. Okay. Yes. The first concept. Just a moment. Okay. Can you see the slide at home? Yeah?
Yes.
Oh, okay. Okay, thanks. The first concept of this chapter is the concept of chemotherapy. Normally, when we say chemotherapy, we indicate the use of particular compounds to kill cancer cells. But this term in the medicinal chemistry and also in pharmacology is wider. We see that we use a chemotherapeutic agent when we want to hit an host organism, an exogenous cells like bacteria, virus. And like a magic bullet, you see, the smart weapons. In this case, we try to hit an exogenous microorganism by exploiting particular features at different pathways, at different biological structures, at different behavior. So, in medicinal chemistry, the chapter of chemotherapeutic agents is very wide because we have antibiotics, antiprotozoa, antifungi, anti-cancers, antiviruses, and so on. We'll start with antibiotics. These molecules are have a low molecular weight, very small. Normally, they are intermediates of particular metabolic pathways. And these intermediates are able to kill other cells. So they are very important for the survival of bacteria in a particular situation. Here you can see a classification of chemotherapeutic agents as, for example, antibacterial, antifungal, antiviral, anti-cancer, and so on. Antibacterial drugs can be divided in two main groups. The first one is represented by natural compounds. These natural compounds can be modified in a lab. So we can start from a natural compound and then we can modify this compound in order to increase its potency. We'll see a lot of examples of this class. Beta-lactamase, tetracyclines, chloramphenicol, macrolides, peptides, and so on. The second class is represented by synthetic antibacterials, such as sulfonamides, dihydrofolic reductase inhibitors, quinolones, and so on. These are the two main classes of antibacterial drugs. But first of all, I want to remember you the problems related to the antibiotic resistance. The wide use of these agents, of these drugs, is responsible for the presence of particular resistant strains of particular bacteria. Here you can see in the picture the situation in two different years. 2009, you see only one strain is resistant. We have 7 effective antibiotics. 2019, only 3 effective antibiotics. And this is the situation. You know what is an antibiogram? This is the situation. Resistant, resistant, resistant. We have a series of different antibiotics on your left. You see all this Pseudomonas aeruginosa is resistant to all antibiotics. And so what can we do in this situation? We can try to put together, to associate 2 or 3 or 4 different antibiotics at the same time. But you can understand, it's not an ideal approach. We need to have new antibiotics. But the question is, why we don't have new antibiotics? Why the different companies do not research, do not try to find new molecules? The answer is related to the period of treatment when you use an antibiotic. Normally, we use it for 10 days, 1 week, no more. Otherwise, if this company has a drug for blood pressure, in this case, the old people use this drug for 6 years, 7 years, 10 years of the whole life. So you can understand the difference. Antibiotics is used only for 1 week, 10 days. Other classes of drugs are used for a long time. This way, they can earn a lot of money in the second case. In the first case, no. So, in my opinion, it is necessary a public support of the research of new antibiotics. This is my personal point of view. Different mechanism of resistance. Perhaps you have already seen this type of mechanism during the course of microbiology, I don't know, but just to remember you, the presence of efflux pumps. So we try to reach the inner part of a bacteria, and then there is a efflux pump able to take your antibiotic and then to put it outside the cell. In addition, we can find degradative enzymes able to metabolize your antibiotic, resistant genes, enzymatic alterations. So you can understand that we have to fight against different mechanisms if we want to maintain an active antibiotic against particular strains. Here in this picture, you can see the resistance percentage isolated in Europe. So you see the two worst countries are Greece and Italy. So in Italy, we have the highest amount of resistant strains. So we have to use antibiotics, but very carefully. It's a potent weapon in our hand, but we have to use it very carefully to avoid the formation of resistant strains.
But there is no specific reason, like why in Italy and Greece?
This is a,
There is no specific reason why just in Italy and Greece.
because in Italy we use a lot of antibiotics. Against flu, we use antibiotics, but it's bad. And so, if the use of antibiotics is general, it's possible to form resistant strain. This is the reason. The use of antibiotics should be controlled by medical doctors. But normally in Italy, oh, I have just a little bit flu and so on, and so I take an antibiotics. Or otherwise, we can start with the antibiotic therapy and we use it only for 3 days and not for 10 days. And this is another reason responsible for the formation of resistant strains. Bad management. Okay. So Other easy concept, bacterial cell wall. You already know the structure of bacteria. Fortunately, the bacterial cells are quite different from mammalian cells, and in particular, you know, they have a cell wall constituted by peptidoglycans. Peptidoglycan is a polymer able to defend the bacterial cell from enzymes, from the acidic condition, from and so on. You know also the difference between gram-negative and gram-positive. Gram-positive bacteria have an external cell wall, very easy to reach. The problem normally is related to gram-negative, because if you remember, okay. Gram-positive, you see the structure, peptidoglycan outside, and then the cell membrane. Gram-negative, there is an external membrane, and then there is a peptidoglycan cell wall, and then another cytoplasmic membrane. You can see two important structures in this picture. The first one are the penicillin binding proteins, PBP. Penicillin binding proteins represent the target of particular antibiotics. We have to reach this type of proteins if you want to kill this type of bacteria. In the case of gram-negative, you see that if we want to interact with this type of proteins, we have to cross the external, the outer membrane, the peptidoglycan layer, another membrane. So it is not so easy to reach these proteins. But fortunately, there are particular channels called porins. Porins are particular channels. They cross all the outer membrane. So if you use hydrophilic molecules, we can use this type of channel to reach the PBP. So we'll see how this strategy was applied to have wide spectra antibiotics. But why we need wide spectra antibiotics? When you have an infection, normally we have no time to make an antibiogram because it's too long. I have to wait 1 week, in the past now 2 or 3 days. And so you stay with fever, with pain, without antibiotic, and so on. So normally, in this case, when you have an infection, we take an antibiotics immediately, but we need a wide spectrum antibiotic because we don't know what kind of bacteria is responsible of my infection. And so we'll see how it is possible to enlarge the spectra of specific antibiotics by increasing the hydrophilicity of particular molecules. So, have you any questions? I think you have already seen a lot of these things in the course of the macrobiology. Just to recall you some important concepts, important for me. Just a glance to the chemical structure of peptidoglycan. As I told you before, peptidoglycan is a polymer, a natural polymer. There are two main chains formed by two different units, repeated units. N-acetylglucosamine and acetylglucosamine. You see the basic group, the NH2, is acetylated. So N-acetylglucosamine joined to another molecule called N-acetylmuramic acid. N-acetyl, once again, you see the amino group is acetylated. And then there is the residue of lactic acid, this position. And the acidic group is bound to a peptidic chain. So we have two main chains with a lot of cross-linking chains constituted by different amino acids. And in particular, you see there are non-natural amino acids, D-ALA, you see, a lot of non-natural amino acids. Why? Because in this case, the peptidoglycan cannot be attacked by our enzymes. Our enzymes are not able to destroy, to digest this type of chains. The most important point of this structure is D-ALA, you see, this alanine, and this pentaglycine bridge, 5 glycine residues altogether. This is an important point because we see how beta-lactamase block the formation of this particular bond in this position. So this is the chemical structure of peptidoglycan. A lot of sugar residues, you see, with these peptidic chains with the non-natural amino acids. So we can stop now and then we start again with the formation of this of peptidoglycan. So have you any questions? No? No? Okay. So we start again 3.5. Okay. So just a moment. Ok, possiamo chiudere qua. Allora. One of the most important points in the biosynthesis of peptidoglycan is the formation of this bond between D-ALA and the last glycine of this pentaglycine bridge. Here you can see an important unit of the peptidoglycan, an unit of muramic acid, you see, and acetylglucosamine and NUDP and acetylmuramoyl pentapeptide. So it is important that you see that this amino acid chain has a particular final tail constituted by two residues of D-ALA. But you can see in the structure, the alanine is only one. What happens in the biochemical synthesis of peptidoglycan? How the pentaglycine bridge can attack the two D-alanine, the two final D-alanine residues. There are important chemical considerations that we have to do. Here you can see the last two alanine residues And here you can see the lone pairs of the amino group of the pentaglycine bridge. And this basic nucleophile is not able to attack directly this carbonyl. Why? Because this carbonyl is not enough electrophilic. Why? Because this carbonyl belongs to an amide group. And you know that an amide group is not so electrophilic. So it is necessary the intervention of a particular enzyme called transpeptidase. Transpeptidase has a serine residue, a negative charge serine residue, a very strong nucleophile, able to attack this amide carbonyl, amidic carbonyl, in order to give this intermediate. You see the diala is bound to the serine residues of transpeptidase. Now you can see we have an ester in this case. No more an amide, we have an ester. And an ester can be considered as activated carboxylic residue, carboxylic acid. And so now the carbonyl of the ester can be attacked by the basic group of the pentaglycine bridge to give the peptidic bond and to have the cross-linking of the two chains. We have a new bond. So you can see the importance of the transpeptidase. This enzyme is able to activate the diala tail in order to have a new peptidic bond. Penicillins are able to interact directly with this enzyme, with the transpeptidase. You can see here what happens. Here you have a 4-member ring, a constrained ring, very reactive, and this carbonyl group looks like the last amido group of the peptidic chain. So the two structures, diala-diala and the beta-lactam ring, are very similar. So the OH group of transpeptidase prefers to attack the more reactive beta-lactam ring instead of the carbonyl group of diala-diala. In this way, you can see the formation of a covalent bond and the serine residue of the transpeptidase is blocked. So penicillin are able to block the formation of this particular bond between the two chains of peptidoglycan. This means that this ring is very reactive. This carbon atom is a strong electrophile that can be attacked from a strong nucleophile as a serine residue. Once again, here you have a summary scheme. You see the negative charge serine residue, O minus, oxygen minus. You have the natural substrate, diala tail. Once again, you see the O minus is able to attack this carbonate. If penicillin is present during this step, also the beta-lactam rings can be attacked by this nucleophile. In this case, we have a complex penicillin enzyme. So these enzymes, this complex represents a block in the pathway of the biosynthesis of peptidoglycan. Now, it is important to understand why the beta-lactam ring is so reactive. And so we can start to analyze the chemical features of the beta-lactamans. Beta-lactam ring, you see, is a very constrained ring. 4 elements. One is a cyclobutan 2-O, you see. But what means beta-lactamans? Beta, because there are 2 carbon atoms between the amido function. If there are 3 carbon atoms, we have a gamma-lactamans. We can have delta-lactamans. It depends on the number of the carbon atom present between the two, the carbonyl and the nitrogen atom. Here you can see the main backbone, the main structure of penicillin. This structure is called 6-amine penicillinic acid. Amine because you see there is an amino group in 6th position. Acid because there is a acidic residue on the carbon 3. Penicillinic indicates these 2 rings fused together. We have a beta-lactam ring and another 5 atoms ring called thiazolidinic ring. Thia means sulfur, azo means nitrogen, thiazolidinic because it's saturated in all positions. So penicillins, the main unit, the most important backbone, the most important part of the penicillin is constituted by this structure, aminopenicillic acid, aminopenicillamic acid, sorry. So at this point, we have to see how many carbon atoms are chiral. You see, we can start from a sulfur 1, carbon 2, no, because there are two methyl groups. Carbon 3 has 3 different substituents. So carbon 3 is a chiral carbon atom. Nitrogen 4, no. Carbon 5, yes. You see, there is the hydrogen and then the other 3 important atoms. Carbon 6, once again. So we have 3 chiral carbon atoms. Only the 3s, 5r, 6r is active. All the other isomers are not active. So these 3 stereocenters are very important. Here you can see the model of the aminopenicillinic acid. You see here in red the acidic function. Here you can see the 2 methyl groups. This is the sulfur, this is the thiazolidinic ring. Beta-lactam ring, you see 4 atoms. So this is carbon 3, carbon 5, and carbon 6. It is important that these 2 hydrogen atoms remain under the plane in alpha position, you see. And it is important that also the acidic group remain under the plane. So the acidic group and the amino group are in opposite position. But, okay, once again, you can see the IUPAC name, it's not so easy, but fortunately we have ChemDraw, just a moment, so we can show you ChemDraw here, okay. It works now, just a moment. Okay. I share my screen. Here, here, and here. Okay. So, we have this type of ring. Yeah, this type of ring. So, and okay, we can put here sulfur atom here, and here we can put COH, and here NiH, and then we have N and then this one. Okay. So we can see now the chemical name. Just a moment. Okay. You see the name here, the chemical name. 6 amino, 7 oxo, 4 tia, okay. 1 asa bicyclo, 3,2,0 heptane, 2 carboxylic acid. What means this part? 1 asa bicyclo, 3,2,1 heptane. Heptane, please. 1, 2, 3, 4, 5, 6, 7. So there are 7 atoms which compose these 2 rings. Heptane. But we can draw another type of heptane. This one, for example. You can see the name. Bicyclo 2,2,1 heptane. Which are the differences between these 2 bicyclo systems? These 2 atoms are the bridgehead. These 2 atoms are common to the 2 rings. You see the nitrogen and the carbon in this position. In this case, these 2 carbon atoms are common. In this case, we have 1, 2, 3 atoms on your right, 2 atoms on your right, and 0 atoms between the 2 bridgehead atoms. 3, 1, 2, 3, 2, 0. In the second example, you have 2 on your right, 1 and 2, 2 on your left, and 1 in the middle between the 2 bridgeheads atoms. 2, 2, 1 heptane. So you can understand the chemical name and how we can define this ring system. In this case, you have 2 fused rings without any carbon atom between the 2 bridge heads. In the second case, you have 1 carbon atom between the 2 bridge heads. So when you see bicyclo, you have to imagine a system of the 2 rings, and you have to read carefully the number, the square. Is it clear? Yeah? Okay. Now, is it clear at home? Yes, no. Okay, check, perhaps. Okay, okay, thanks. Allora, qua. So we have explained the chemical name of this system. And now we have to understand why this amide is reactive and why this amide is not reactive. Why the beta-lactamase ring is so reactive? So, you know that a nitrogen atom has a lone pair, basic lone pair. In an amido group, this lone pair is shared with the carbonyl atom, is delocalized. And so you see here the two resonance forms which describe the distribution of the two electrons inside the amido group. This means that the amino bond is very strong, is almost a double bond, and so is not so reactive. If we see the behavior of the nitrogen atom inside the aminopenicillinic acid inside this system, we see that the distribution of the lone pairs is not possible. So the lone pairs remain to the nitrogen atom, on the nitrogen atom, cannot be distributed, delocalized on the carbonyl system. You see, it's not possible, the delocalization of this lone pair on the carbonyl group. This means that you have a carbon atom between an oxygen and a nitrogen. Oxygen and nitrogen atoms are more electronegative than carbon. This means that you have a strong positive, partial positive charge on the carbon of the beta-lactamase ring. If you make a Nair spectra of a penicillin, you see that the frequency of this carbonyl is quite the same of an activated carboxylic acid. Activated means that you put a chlorine on the carboxylic acid structure. Also in this case, you have a carbon between two more electronegative elements, an halogen and an oxygen. This means that you have a very reactive electrophilic carbon atom. So, when during the exam I ask you why this carbonyl is so active, you have to explain this situation, that the distribution of the lone pairs is not possible, and so this carbon atom is very electrophilic. In the other case, when you have a normal amido group, like in diala tail, this carbon is not so reactive, because you have the distribution, the delocalization of the lone pair. But why in the beta-lactam ring is not possible the delocalization of the lone pair? Once again, we have to observe the tridimensional structure of the aminopenicillinic ring system. Sorry. You see here the two rings. The two rings are not planar, but they are almost perpendicular. This means that the nitrogen atom is not able to delocalize its lone pair on the carbonyl because they are perpendicular. We say simil sp3 carbon atom is a nitrogen, but in the normal amido bonds, the carbon is sp2. So it's quite different. So this is the explanation of the different behavior of the beta-lactam ring in comparison with a normal amido group. Is it clear for you? Have you any questions? Chemical questions? No? So this is the reason why this four-term ring is just so reactive. No delocalization of the lone pair of the nitrogen atom. There is another important family of beta-lactam classical beta-lactam derivatives. The second family is called cephalosporins. We can analyze the chemical structure of cephalosporins. Once again, you can see the four-term ring, beta-lactam ring, but now we have a six-member ring fused with beta-lactam. So this six-member rings is called dihydrothiazine ring. Dihydro, because we have only an unsaturated bond instead of three, if you know, on the benzene, for example, only one. Thia is related to the presence of sulfur, And so penicillins are characterized by thiazolidine ring. In this case of cephalosporins, we say the hydrothiazine system. You can see the presence of this atom, X. This atom is a good leaving group. In the case of cephalosporins, we need an optimal leaving group. If you remember from organic chemistry, good leaving group are halogens with a minus charge, or halogens, or acetic acid, ionized acetic acid, the acetyl residue, and others, because we need a good leaving group in this position. Once again, we can analyze the chemical name of these two ring systems. We have a beta-lactam, 4 components, 4 atoms, and then we have a 6-member ring. In this case, we say one has a bicycle 420, octa 2 in. So why 4? 1, 2, 3, 4 on your right, 4. 2 on your left, 1 and 2, and 0 between the two head bridge atoms. Bridgehead atoms, sorry. This is the reason of this name. You see, in because we have a double bond in the cycle. If we see the model of the cephalosporins, yes, okay. You see the sulfur is here, the leaving group is here, the acidic group is here, and then the 4-ring system here, okay. The 2 rings are planar. The situation is different from that of penicillins. So the reactivity of cephalosporins is not due by geometrical condition, but for the presence of the double bond and the leaving group. We'll see this important point later when we describe the chemical structure of cephalosporins. Yeah?
For the presence of double bond
And the presence of the leaving group. We'll see the importance of the leaving group in the cephalosporins to increase the reactivity of this type of system. How penicillins and cephalosporins are biosynthesized inside the microorganism by the different microorganisms. There are 3 important building blocks, 3 important brick, okay, we can say, L-cysteine, an amino acid, L-valine, another amino acid, and another atypical amino acid called L-aminoadipic acid. Okay.
L-aminoadipic acid.
Adipic, yes. You see, the chemical structure is like an amino acid, an acidic amino acid, because glutamic acid, for example, is quite the same. It's not so common. And the different microorganisms are able to put together, to organize these three important compounds to get the structure of penicillin. So the first step, the first part of this pathway is related to the formation of the penicillin N. After that, there is an expansion of the thiazolinidine ring, of the 5-member ring, to 6-member ring. And in this way, we have cephalosporins. So cephalosporins derive from penicillin. But why there are some strains resistant to this type of antibiotics? Because there are 3 different ways of inactivation of these 2 families, penicillins and cephalosporins. The first one is due by the presence of beta-lactamases. These proteins, these enzymes, are characterized by a basic groups, a basic residue. This basic residue is able to attack the reactive carbonyl of beta-lactamase and to open this ring. The result is an inactivated penicillin. There is another class of enzymes able to inactivate penicillins and cephalosporins. This second class, second family is called amidases. Amidases are able to hydrolyze this amino group present in the side chain of penicillins and of cephalosporins. They hydrolyze an important part of the side chain, and this side chain is necessary to have a strong interaction with transpeptidase. So you can see in the figure, beta-lactamases are able to open the 4 atom ring here, this point, on this carbon atom. The amidases are able to hydrolyze part of the side chain. And finally, there is another family called esterases, able to hydrolyze the leaving group of cephalosporins. As I told you before, normally optimal leaving group is constituted by an acetyl residue, and so we have an ester in this position, and this ester can be hydrolyzed by esterases. These three families are able to inactivate penicillins and cephalosporins. The most important family is represented by beta-lactamases. If you want to protect the activity of penicillins and cephalosporins, we have to apply a strategy in order to defend the beta-lactam ring from the attack of beta-lactamases. Now we start with the description of penicillin. Perhaps you already know the history of penicillins. They were discovered in 1928 from Fleming's observation. In that time, Fleming went to holidays in August. And during August, he didn't close the window of his lab, his laboratory. At the end of the holidays, he came back in the lab and he observed this situation. The formation of particular inhibition halogens around the bacteria colonies. And for a scientist, normally, when this situation happens, normally we start again with all the experiments, and we do not try to understand the reason of this phenomena. But Fleming was different, thought differently. He tried to understand why the reasons of this phenomena. And he repeated again the different experiments. And he discovered the presence of particular compounds able to block the growth of bacteria. But the problem was really difficult to solve. Why? Because the chemical structure of these compounds were in that time very uncommon. And in that time, they had no equipment no experiments to determine the chemical structure of penicillin. And also penicillins are very unstable. And so you can understand in that time was really a big challenge, the determination of the chemical structure of these compounds. However, you see, 1940s, demonstration of the therapeutic ability of this compound. 1945, X-ray determination of the benzyl penicillin structure. 1957, isolation and characterization of the nucleus of penicillin, 6-aminopenicillamic acid. In 1977, total synthesis in lab. Nobel Prize, Sheehan and collaborators. Because it's not so easy to synthesize, but they synthesize this compound in lab to demonstrate their chemical structure. Some years later, it was possible to have pure 6-aminopenicillamic acids, the starting point of new penicillins, by changing only the side chain. So, if we have pure 6-aminopenicillin acid, it's possible to synthesize a large number of new penicillins. How? It's very easy. You have an amino group in this position. You can have an activated acidic group with an halogen, for example, or with an anhydride, and then you have a new penicillin. You see here the different nature of the substituents in the side chain. Here in this picture, you can see the similarity of the chemical structures of the diala tail and the similarity with the beta-lactam ring. Just to understand why penicillins are able to compete with the same site of the transpeptidase, the same site of the diala of the transpeptidase, the active site of transpeptidase. So, have you any questions at this point? Have you any question at home? No? Okay, we need time to explain this point, this synthesis, and so perhaps I can describe this synthesis in the next Monday. And here I want to show you how to have new penicillins. You see the basic nucleus, 6-aminopenicillinic acid. We have to use activated acids, organic acids residues, with different methods, just to describe you, but I don't ask you this method during the exam, just to show you different ways to have activated acidic residues to put together with 6-aminopenicillinic acids. You see different approaches. Now, I'd like to describe you the development of new penicillins during the last years. The first penicillin discovered by Fleming and co-workers was benzyl penicillin. You see here the chemical structure, you see the R substituents here. In our case, R is represented by a phenyl and an amethyl group. Benzyl, this group is called benzyl penicillin, called also penicillin G. Penicillin G was isolated by Americans in USA during the Second World War. This molecule, penicillin G, has a lot of problems as a drug. It is stable only at physiological pH, 7.4. It is active only towards gram-positive. Very restricted spectrum. It should be administered only by injection, not orally.
Just to be sure, we're talking about penicillin G.
G, yes, the first penicillin on the market. 44, 45, 1944, after the Second World War, during the last part of the Second World War. And this penicillin it is not stable towards beta lactamases. But nevertheless, it represents the first weapon against infection. In that time, we had only sulfonamide, a very old class of antibacterial agents, and not so strong. And penicillin G represented really a revolution in the therapy of the infections. The first point in that time was to use this penicillin orally. And in that time, they introduced an oxygen atom, you see here, between the aromatic ring and the carbon atom. And we get penicillin V. phenoxymethylpenicillin, penicillin V. So the first was penicillin G, and the second one was penicillin V. This penicillin is stable at acidic pH. So it was possible to administrate this compound orally. And this is really another revolution. We'll see during the next lesson the reason of this behavior. Why penicillin V is resistant to acidic pH. We'll see the chemical reason. The introduction of the oxygen is important to stabilize this compound at acidic pH. After these two important penicillins, yes?
The question to the penicillin V, it's the introduction of an oxygen atom, right?
Yeah, you see here in this, you see here, oxygen atom. And this introduction is sufficient to stabilize the backbone of penicillin at acidic pH. Brilliant. So we can use Phenospen, is the market name. Phenospen is stable at acidic pH. After these two important penicillins, other were developed, but they are not so important as the two first penicillins, G and V. After these two important penicillins, the scientists try to introduce some bulky substituents on the aromatic ring. Why? Because in that time they tried to find to stabilize our penicillins to the activity of beta-lactamases. But why bulky groups on the aromatic ring? You can see now in the picture, we have a bulky group, a large electronic clouds with a negative charge, very close to the beta-lactaman ring. And this ring is the target of beta-lactamases. So the beta-lactamases has a nucleophilic group with a partial negative charge. And this group should attack the carbonyl of the beta-lactaman ring. An electrophilic group has a partial negative charge. And so if there is a negative clouds very close to the beta-lactaman ring, we can protect this ring by the attack of the beta-lactamases. The two clouds have the same charge, and so they are repulsive. This is the reasons why in that time we can find several penicillins with bulky substituents on the aromatic rings, or with the addition of heterocycle in the side chain in order to form a large electronic clouds to protect this part of the molecule. Yeah.
So first, were these bulky groups responsible for extra side effects in these cases?
No, only to protect the beta-lactam group, no side effects, no. This type of penicillins were active, but they try to form a clouds just around the beta-lactam ring to protect it from the attack of the beta-lactamases. Beta-lactamases is not the target. The target is transpeptidases. Beta-lactamases are enzymes able to open the four-member ring. And so if you want to defend your four-member ring, you have to form a cloud with the same charge of the beta-lactamases. And so you have a repulsion of the two entities. It's clear?
Yes, and another more like curiosity, how much after the commercial being available commercially, penicillin did it take for the bacteria to be resistant?
I don't understand.
How much after penicillin was commercially available for the people, did it take for the bacteria to form resistance?
I don't know. No, I don't know. The resistance in bacteria appears during the time. And so it depends of the use of this type of antibiotics. And if you use this compound correctly for 10 days, for example, for 1 week, for example, you kill all type of bacteria, and then you have not resistant strains, the formation of a resistant strain. If you use this type, there is another problem, yes, okay. We eliminate a lot of these compounds by urine. Secondly, a lot of these compounds are used in agriculture to protect animals like cow and so on from the infection. And they use low concentration of these compounds for a long time. This is the best way to get resistant strain, the formation of a resistant strain, when you use low concentration for a long time. So if you put together the quantity of these compounds eliminated in the river, in the sea, and so on, together with the wide use of this compound in veterinary agriculture and so on, you have a synergistic effect in the formation of a resistant strain. This is my opinion.
No.
Yeah?
I just have a question. These bulky groups that are usually added before, it's still used now, this technique?
Yeah, really. We see the same strategy in cephalosporins. Yes. A negative cloud around the beta-lactam ring. Yes. Other questions? At home? Okay, dear students. I think we can stop now. So next time, next Monday, we start from this synthesis. So we have enough time to explain it. It's very interesting. And so you can understand how we can obtain a pure 6-aminopenicillinic acid. It's very, very, very important synthesis, yes. Okay. So, dear students, thanks a lot for your attention of today. Also at home, thanks a lot.